So Marko Pende is a postdoc in the Merwala lab. Did I say that right? I always get, no, he'll fix it. It's just, okay, all right, he's not here, so it doesn't matter. He has 2 Masters of Science degrees, one in molecular biology and one in immunology from the University of Vienna, and a PhD in biomedical engineering from the Technical University of Vienna and the Center for Brain Research, Medical University, also in Vienna. In 2018, Marko received the Konstantin von Economo Young Investigator Award from the Austrian Neuroscience Association. And then just to remind you all that these folks are normal human beings like the rest of us, I asked them to let me know what their favorite pop culture science thing is, which it's just fun to ask what it is. So Marko's was, his favorite science themes books are the Foundation series by Isaac Asimov and Sapiens, A Brief History of Humankind by Yuval Harari. And Marko's talk is 3D microscopy his talk title. See, I can't even do it. You guys are going to be so much better than I am at this. His talk title is 3D Microscopy Unlocks New Ways to Shine Light on Biology.
Yes?
Yeah.
Now I can start. Okay, perfect. Welcome everybody to this exciting evening and welcome to my talk. Thanks, Kate, for inviting me here. I'm very honored. And yeah, let's dive into it. So, very early microscopists, pioneers in this field, like Ramonika Har, who you can see here, used, and he basically received the Nobel Prize in Medicine, had worked with very thin sections on slides and observed the sections with these microscopes. Unfortunately, at this time, cameras were not invented, and so he had to draw his observations. Microscopes became more evolved and became more sophisticated. However, we still have these histological sections where we observe the biology. And what you can see here is a neuron. So we have here the cell body and you have the projections going away from the cell body. But neuron is not a 2D structure. Actually, you have processes going in all spatial directions. And this neuron is also surrounded by other neurons to which it is connected to. And when you think about a mouse brain has 8 to 14 million neurons. And you cannot see these things on such a picture. So, keep in mind, biology is in 3D, and when you show it in 2D, you can miss actually crucial information. Another thing is that these microscopes are built for very tiny samples. And even if you can fit a mouse brain inside such a microscope, because the sample is not transparent, you can just observe the most outer layer of such samples. And here I actually have an actual mouse brain. Now, nature helped us out and inspired us, and I'm also very inspired by these animals. These animals actually really exist, and they're not photoshopped. So with these animals, you don't have to do many sections. You can just look inside. And so I was, or many scientists were asking themselves, can we actually do our samples transparent? And to do so, we have to understand how transparency works. And here in this flask, we have air and water bubbles, and by removing the air, actually you can see suddenly the picture of Werner Spalteholz, who is the father of a technique called tissue clearing. And here you see a picture of a cell, and when you think about the cell, it consists of water and different cellular components like mitochondria, Golgi, lipids, and we and other organisms consist of many cells. And by doing some chemical tricks, we can actually make from such a not transparent sample a transparent sample. Now, I mentioned sections. We have a transparent sections, and we cannot fit the sample inside of the usual microscope. So we had to build our own microscope. And here you see like a chamber with the tissue-cleared brain, which has GFP signal inside. And this is the chamber. And yeah, this is a so-called light sheet microscope. And this works that you make from a point source of a laser a light sheet. And this sheet kind of scans through this transparent sample, which you can see here. And this is actually how it looks like. It's a bit distorted right now. And these are the images which we actually generate with the computer. And by having many, many of such planes, we can actually generate a 3D image. And every dot you can see here represents actually a neuron. And now we can look at the entire brain from all spatial directions. And if you wait a couple of seconds, I still have time, then we can even fly inside the brain. Wow. Yeah, the current applications at the MDI-BL, so we want to understand the neural networks to decipher brain function. We use these techniques for tissue regeneration after cutting of limbs and for visualization of kidney vasculature for drug targets. And with this, I thank my group and the light sheet microscopy facility. 1 second. And Frederick. And I'm happy to take your questions.
Coming up.
Hey, long-time listener, first-time caller. So I'm intrigued by the fact that you're using point clouds to represent three-dimensional data, but you're representing them still in a two-dimensional interface. So are you using, if you thought about using anything like virtual reality goggles or augmented reality goggles to actually experience the data in 3 dimensions by parsing it from 2D still into 3D?
I mean, for example, people in Australia did it. I'm not sure which lab, but they call it the pit, like we call it here, actually. And yeah, I send them some of my stacks, and they actually do kind of have a projection room where people can move inside the brain. We didn't do it yet. I mean, it would be very nice, and I have actually VR goggles on my own, so let's go play games.
How long did it take you to build the microscope?
From getting the funding, 6 months to order everything, 2 days to build it.
Are you going to tell us how much it cost?
$500,000. This is cheap.
Yeah. Last chance. Oh.
So how long does it take to take an image?
For such a brain, it took me 10 minutes.
So can you do time resolved? Can you watch things evolve over time through it?
Beg your pardon? Where are you actually?
Can you do time resolved? Can you watch things evolve over time?
Yeah, with our techniques, I mean, I use tissue clearing, so the samples are dead. But if you have a transparent sample like a transgenic zebrafish, yes, you can do. Definitely. Actually, one of this very early scientists who did light sheet microscopy did this on zebrafish. And commercial companies developed also a light sheet for zebrafish size, for example.
So what's your fantasy organism to use this on? What would be the ideal thing for you to do?
My fantasy.
You can dream, just dream.
Actually, axolotl is quite cool, which I'm working right now with. And I would like to do live imaging with axolotl as well. So I study nerve regeneration, currently in the Moravale lab. So I would like to do a live time lapse on a nerve innervating actually the limb. So this would be pretty, and actually this can happen within 1 week. So it's very possible, and they're semi-transparent. You cannot see it properly right now. Oops. Yes, now you can see it properly. So this is now I cannot see it anymore. But these are nerves which are visualized in axolotl. So I can do live imaging. But I didn't do it yet with light sheet. I mean, they have to be alive and don't move for at least 1 week. So I have to put them like in a coma-like state. It's possible.
All right, go ahead. Yes. Yeah.
What is the biggest thing that can fit in this microscope?
Depending, so I would say something the size of 10 centimeters, depending on the chamber. In the end, it's just the amount of data, and the working distance of the objective is sufficient to image an entire mouse. So you have to do tiling and then stitching later, all these little tiles. And this is quite a substantial amount of time and working memory on computers. But it's basically kind of restricted by the movement of the stage and the working distance of the objective. You can fit an elephant inside, in theory, if you would be able to tissue clear it. And I'm not working on elephants.
Thank you.
Okay, thanks.